Many other syndromes may still be out there and have never been identified before. So it's good to pursue this research as well. So both CDH plus and isolated CDH have more than one underlying cause, and many different techniques need to be employed in order to discover what's going on. Now, I'll show you some of the techniques we use in the lab so that you can sort of appreciate the details of the study that we are running. This is a very common exam. It's called the Standard Karyotype, and it's performed on peripheral blood samples. It's also performed usually during amniocentesis or chorionic villus sampling. So basically, you take a picture of the chromosomes and you line them up, one next to the other. There's two of each kind. One comes from the father, one comes from the mother, and you stain them in those darker and lighter bands that help us define their structure. Because when we look at the karyotype, what we want to know is that the number of chromosomes is right, and there are no structural rearrangements. The standard karyotype helps us identify rearrangements, so structural changes, in about 10% of CDH babies. And these are the structural rearrangements we're more concerned with. They are deletions and duplications. This is a cartoon of a chromosome. Now take a look at the yellow band, because the yellow band might be missing, and this is called a deletion, and the chromosome is shorter and just misses a bit of genetic material. And sometimes the band is duplicated, so we have twice as much as it would be normal. And since the number of genes and genetic materials is so fine-tuned in the human being, both when there is too little and there is too much, that can influence the origin of a genetic syndrome. This is another technique to detect rearrangements and deletions at a very, very small scale. So when you wouldn't be able to, the last two pair of chromosomes looks normal on the karyotype. But if we have a strong suspicion based on clinical data that there might be a deletion in that specific band, we can do a special analysis in a lab. We create a fluorescent probe that detects just a stretch of DNA, and we hybridize it to the chromosome. If we have two signals, this means it is normal. If we just have one signal, this means there is a deletion that is so small that we can't detect it on the normal karyotype. So it's good if a karyotype looks normal, but we have a strong suspicion about a specific deletion in a certain point of the genome, we can run a FISH analysis to be able to detect that. And this is what the FISH analysis looks like under the microscope. You see that this chromosome here, this is the subtelomeric signal that its homologue has. So that's a very, very small deletion. So we can use FISH to detect specific regions in the chromosome. But now there are new technologies that we're also using that allow us to scan for deletions or duplications that are very, very small, and you can run those tests across the entire genome. That is called array comparative genomic hybridization, or ACGH. So we started from the chromosome, the layer, and there's another layer of analysis, which is looking at the genes, specific genes that are made up of a different order and sequence of nucleotides, and reading each nucleotide one after the other, just we would read the letters in a book. So you see each one of these peaks is a different nucleotide. And we can compare the findings in a CDH baby and in reference DNA, that is DNA from a person that is not affected by CDH. And for instance, you can see that that position where we should have a G, we actually find an A. So not only very, very large deletions or duplications that encompass several genes, maybe 20 or 30 genes, or just one gene can cause CDH, but also sometimes very, very small changes. It's enough to change one nucleotide, that is one letter, to cause CDH, if that is in a specific gene that is associated with CDH. So CDH has many different causes. It can be isolated, or it can be part of a syndrome, and it may run in families. The risk of having a second affected child actually depends on the cause of CDH. If we can't find the cause for CDH, as it is for isolated cases, we have to make an estimate based on our previous experiences and what we have seen in the past. And the recurrence risk, the recurrence estimate for isolated cases is about 2%. So now Dr. Russell will tell us about our study design and what we are doing in Boston.
So on behalf of myself and the CDH study staff in Boston, we feel extremely fortunate and grateful for the opportunity to have worked with so many CHERUBS families over the years. And it's been really wonderful to actually meet some of you guys in person, to talk on the phone and all of that. But this is one of the first little girls that we enrolled into CHERUBS. And her mom tells me that she's faced many hurdles and challenges over the last 7 years, but she's doing really great now. And they actually live on a small farm in South Africa, and she's doing great. So some of the main priorities of our study, first is to determine which gene or gene pathways that when they're perturbed or when they're not working appropriately result in CDH in small lungs. We hope in the future that we can design some types of effective but safe therapeutics that we can give to babies, or in utero we can give to moms while they're pregnant, to enhance lung growth before birth, because it's thought that even a small enhancement of the lung growth before the babies are born might decrease the requirement of ECMO and the harsher types of therapies. We ultimately hope to improve care and outcomes for the next generation of babies born with CDH. So we're proud to say that since 2001, we've enrolled families from 48 states, from 25 different countries, and from 6 continents. And the stars represent families that live in all the geographic locations, not necessarily the numbers. So every family that we enroll is considered extremely important, and each family provides a great deal to our study independently as well as in a group, because we do study families alone as well as in combination. So far we have enrolled 365 affected individuals or individuals with CDH. We've enrolled 670 unaffected family members, and among these families, 30 of them are multiplexed, or they have more than one affected family member with CDH. We've also enrolled 7 sets of identical twin pairs, so we say that they are discordant for CDH, or one of the kids has CDH and the other one does not. So studying identical twins has been really helpful, because it sort of lets us into a whole other side of genetics, where we think that identical twins have the same DNA at the nucleotide level, at the base level, the AGCT, but now we're looking at other ways they can be genetically different.